Assalamu alaikum everyone, this is Abdullah Khalil and this is the platform where you learn O-level, A-level and IGCSE biology of various UK boards. Today we are going to start the first chapter of A2 biology, which is called energy and respiration. On our previous knowledge, we know that respiration is a process in which energy is produced within the cells in the mitochondria. The food is being oxidized and the energy is being released. That is respiration. We do not confuse it with breathing. We study gaseous exchange in our AS. So in the A2, this is the first chapter. Again, the first thing that we are going to do is to look at the learning objectives that we have, the learning outcomes as provided by the Cambridge. Now, you can skim through the learning outcomes as given in the notes, and here you will start the first topic. The first topic is, why do we need energy? What is the need of energy in living organisms? See, we need energy for various purposes, and these are listed here. First of all, anabolism. Anabolism is a process. Anabolism is part of metabolism. Metabolism is divided into two types, catabolism and anabolism. Anabolism is the process in which two smaller substances can join up to make one larger substance, or several smaller substances can join up to make one giant molecule or a macromolecule. For example, when the proteins are being synthesized, smaller amino acids will join up and they will make a larger protein. Similarly, when the glucose synthesizes, when the glucose molecules line up to make glycogen or cellulose or starch. So smaller molecules making large molecules is anabolism, and it requires energy. Then we have active transport. Active transport is the movement of substances from lower concentration to higher concentration, and it always requires energy. Osmosis and diffusion do not require energy, but active transport, because the substances are moving from smaller or lower concentration to higher concentration, it does require energy. For example, the ion uptake by root hair cells. Then we have muscular contraction. Muscular contraction enables you to move, to speak, to laugh, to smile. Whatever movement you do, there is some muscular contraction involved in that. For example, breathing, walking, talking, laughing, all those movements require muscular contraction, and muscular contraction requires energy. Then we have cell division and growth. Obviously, meiosis and mitosis, they require energy. Nervous communication. So nervous communication is basically done, it's the electrical impulses that are being transmitted from one end of the neuron to the other end of the neuron. So these electrical impulses or electrical energy obviously requires energy from the cellular respiration. Then we have thermoregulation. That means maintaining the constant temperature of your body. That also requires energy. So these are some of the processes in living organisms that require energy. So energy is really important to make, and it is being made all the time in our body. As we speak right now, there are processes happening in our cells making this energy. The cellular respiration, which is happening in the mitochondria and constantly making energy for everything we do, for all the reactions that are happening, for muscular contraction, for active transport, and for all the other processes that we have just discussed. ATP or adenosine triphosphate. Now this is the molecule, this is the substance that is the universal energy currency. It is used by all the living organisms to supply energy, to fuel the energy requiring processes. Now ATP is the most suitable candidate for this. We could have used glucose directly, but no. Glucose is broken down and the energy is packed into these small energy carriers, ATPs. Why ATP? Because ATP, first of all, the structure of ATP looks like, it is just like the nucleotide of an RNA. Why RNA? Why not DNA? It has adenine, it has ribose sugar, not deoxyribose sugar, ribose sugar, and it has the phosphate group. So it looks like the nucleotide of RNA, and it has 3 phosphate groups attached. So when one of the phosphate groups breaks away, it releases some energy. Because energy, this is the concept that we have to understand, that energy is in that bond. Whenever the bond will be broken, it will release energy. We have studied exothermic and endothermic reactions in chemistry, so we know whenever the bond is broken, the energy is released. So whenever this bond is broken, This is the same way we get energy from glucose or lipids. The glucose has so many bonds, carbon-carbon bonds and carbon-hydrogen bonds. So these bonds are actually the elements that contain energy. When these bonds are broken, the energy is released. This energy is then stored in the ATP, the bonds of adenosine triphosphate. When these bonds will be broken, they will again release energy. But why we use ATP? Because it can be easily broken down. We can easily hydrolyze ATP, we can easily make ATP. When one ATP is broken down or one phosphate group is removed, it releases 30.5 kilojoules of energy. Another phosphate group removed, same amount of energy is released. It becomes AMP. ATP, then ADP, adenosine diphosphate, then AMP, adenosine monophosphate. Finally, the last phosphate group is removed. Now, this time it releases 14.2 kilojoules of energy. These are the 3 points that I have mentioned here, why we use ATP as the energy currency, or why it is the most suitable candidate for this. Because it can be quickly hydrolyzed, because the energy released is not so much that it will be wasted, and it is not so small that wouldn't be enough for the energy requiring process. So it is enough to carry out the process that requires energy, and it is not so much that some of the energy will be wasted. Then ATP is a stable molecule. It requires an enzyme ATPase to break the ATP down. Then we have cellular respiration. Now, what we have studied in IGCSE or O level or up till now is an equation. Glucose reacts with oxygen to produce carbon dioxide, water, and energy. But this is not how it goes. This is just the net equation of what is happening in the cell. Actually, the cellular respiration takes place in 4 steps. So the first step is glycolysis. Glycolysis, the word lysis means breakdown, as we have heard about photolysis or hydrolysis. So lysis is breakdown. Glycolysis is the breakdown of glucose. Then the second process that we have is link reaction. This is the summary that you can see here. The second process is link reaction. Then we have a Krebs cycle. And lastly, we have oxidative phosphorylation. Oxygen is only used in this last step. Oxygen comes into play in only this last step, oxidative phosphorylation. So what happens here? This is the summary. You can see everything that is happening. This is a mitochondria, as you can see, and outside the mitochondria, the first step of cellular respiration takes place. Outside the mitochondria is cytoplasm. If you imagine a cell, you know that outside the mitochondria is cytoplasm. So glycolysis, the first step of cellular respiration, is happening in the cytoplasm. The end product of glycolysis is pyruvate. Once pyruvate is formed after glycolysis, this pyruvate enters into the mitochondria. We know that mitochondria, because we have studied this in our AS first chapter, that mitochondria has 2 membranes. The outer membrane is smooth and the inner membrane has foldings like this. These are called cristae, and there is a fluid present inside which is called mitochondrial matrix. Matrix, just like the movie Matrix. Okay, so the pyruvate that is being formed by glycolysis, this pyruvate enters the mitochondria. Inside the mitochondrial matrix, the next step will happen, which is link reaction. The end product of link reaction is acetyl coenzyme A. This acetyl coenzyme A enters the Krebs cycle. Now, it is this cycle, so there is no end product, but there are substances being made constantly, NAD and FAD. NAD is nicotinamide adenine dinucleotide, FAD is flavine adenine dinucleotide. Now these two act as hydrogen acceptor. Throughout the process, as we will go through, you will see that these two are the hydrogen acceptors. They are going to accept the hydrogen that is constantly being released by different processes, in different processes of cellular respiration. So whenever they accept the hydrogen, they become from NAD to reduced NAD, or from FAD to reduced FAD. So these two steps are happening in the mitochondrial matrix, link reaction and Krebs cycle. Lastly, we have the oxidative phosphorylation, and this is happening here. the inner membrane of mitochondria, not the intermembranal space, inner membrane of mitochondria. This is where the oxidative phosphorylation will happen. All of these processes, the purpose of all these processes is to make energy, to make ATP. Okay, just keep that in mind. Okay, moving forward. Now, to understand glycolysis, we have studied in biological molecules the structure of glucose. Okay, we know the structure of alpha glucose and beta glucose. But for this, just consider the central skeleton, the carbon-carbon bonds in glucose. There are 6 carbons attached just like this. So there are 6 carbons which are called, which is basically glucose. But as the glycolysis begins, the first thing that is going to happen here is one ATP will lose its phosphate. The purpose was to make ATP, but actually we are using ATP here. So ATP is being used here. It will release phosphate and it will become ADP, adenosine diphosphate. This phosphate attaches to one of the carbons, one of the end carbons of the chain, and this becomes fructose phosphate. We know that glucose and fructose they both have basically similar molecular formula. The structure is different, but they have the similar formula, C6H12O6. This is the formula for glucose as well as fructose. So it becomes fructose phosphate, more symmetrical. Now, another ATP will release its phosphate group. Again, the same thing will happen. The phosphate group attaches here and it becomes 1,6-fructose bisphosphate. So this is 6 carbons and each end they have phosphate group attached. So this is 1,6-fructose bisphosphate. 2 ATP molecules have been used. Now this is broken down. This 1,6-fructose bisphosphate will be broken down from the middle and it will make 2 symmetrical triose phosphate molecules. So we have 2 triose phosphate molecules. These 2 triose phosphate molecules will release 2 ATP and 1 H when they are broken down into intermediates. So the 2 ATPs that we have used in the beginning have been recovered here. Okay. Now we have 0 net gain of ATP up till this point. One of the H that is being released, the hydrogen that is released, it reduces the NAD and NAD becomes reduced NAD. This reduced NAD is very important. We are going to collect all the reduced NADs and they will be used in the last step. So always keep that in mind. This reduced NAD is really important, more important even than glycolysis in terms of energy production. So reduced NAD is being stocked, so it will be used at the end. The intermediates will again be broken down and they will release 2 ATP and the end product is formed here, pyruvate. Obviously, because we had 2 triose phosphate molecules, so 2 pyruvate molecules will be formed here. Each pyruvate will have 3 carbons. This is the end product of glycolysis. So glucose becomes fructose phosphate and then it becomes fructose 1,6-fructose bisphosphate. That is broken down into 2 triose phosphate molecules. They release ATP, they make reduced NAD by releasing hydrogen, and eventually they are converted into pyruvate. The net gain of ATP is 2. Why? Because 2 ATPs are used here and 4 ATPs are produced here. So the net gain is +2. Let's move on. Now this has happened, if we remember, this has happened in the cytoplasm. The pyruvate is formed in the cytoplasm. But once the pyruvate has been formed, it will enter in the mitochondria. Just like this. So pyruvate will enter in the mitochondria. What will happen next? When the pyruvate enters the mitochondria, 2 processes will happen. First, decarboxylation, that is removal of carbon dioxide, and dehydrogenation, removal of hydrogen. And we know hydrogen, what will happen to this hydrogen? Accepted by NAD and it will form reduced NAD. So we have reduced NAD being formed here and decarboxylation is happening. That means carbon dioxide is removed. Now the pyruvate was 3 carbon substance. It had 3 carbons. Once a carbon dioxide is removed, it becomes a 2 carbon substance. This will react with coenzyme A and it will make acetyl coenzyme A. So this would be the end product. The net equation, as you can see here, pyruvate is being used here, coenzyme A is being used, and NAD is being used. NAD becomes reduced NAD by accepting hydrogen. Carbon dioxide is released by decarboxylation, and because the remaining substance will react with coenzyme A, and it will make acetyl coenzyme A. The end product of link reaction is acetyl coenzyme A, which will now enter the Krebs cycle, happening again in the mitochondrial matrix. What happens in the Krebs cycle? This is important. Now link reaction releases this acetyl coenzyme A, and this acetyl coenzyme A reacts with oxaloacetate already present here. Oxaloacetate is a 4-carbon compound. As it reacts with acetyl coenzyme A, it makes a 6-carbon substance called citrate. That's why Krebs cycle is also called citric acid cycle. Citric acid cycle, because it was discovered by Sir Hans Krebs. So to honor the scientist, the name of this cycle is Krebs cycle. Twice the same thing will happen to the citrate. Decarboxylation, dehydrogenation, it becomes a 5-carbon compound. NAD, reduced NAD is produced. Again, decarboxylation, dehydrogenation, it becomes a 4-carbon compound, and again reduced NAD is produced. Once it has become a 4-carbon compound, it will make the ATP. Now, this time, and this is the only time in all of these 4 processes that FAD is being used. This FAD, flavine adenine dinucleotide, will become reduced by accepting the hydrogen, and it will become reduced FAD. Again, another hydrogen will be released to make reduced NAD here. And finally, this becomes oxaloacetate again, to repeat the cycle, to react with acetyl coenzyme A again to make citrate again. This is a cycle that this keeps on going in the mitochondria, in the matrix of mitochondria. The NADs and FADs that are formed here, they will now move towards the inner membrane of mitochondria. Because it was happening in the matrix, this will now move towards the inner membrane of mitochondria. What happens here? The most important or the most productive in terms of energy, the last step, oxidative phosphorylation. This process is most productive in terms of energy. How? If you look at this, you can see Christie is labeled here, or the inner membrane. If you enhance the size of this, you will see something like this. Because it is a phospholipid bilayer, the Christie is a phospholipid bilayer. So you will see a bilayer here. This is phospholipid molecules. I have not drawn it completely, but just for the understanding, these are the phospholipid molecules. It would appear like this. I've just drawn the lines to understand. Reduced FAD and reduced NAD will release their hydrogen here, will lose their hydrogen, and this hydrogen will be broken down into, because hydrogen has only 2 things, the proton and the electron. Hydrogen will be broken down into the proton and the electron, the H plus and the electron. Electrons are then transferred to electron carriers that are present here, and it will be transferred from one electron carrier to another electron carrier, and then to another electron carrier. This makes electron transport chain, okay? And in the electron transport chain, the electron is constantly releasing energy. That energy is used to transfer this H plus out of the inner membrane. When it goes out of the inner membrane, it goes into the intermembranal space. In between the inner membrane and the outer membrane of mitochondria, the H plus is stuck here in between. And it is constantly happening with all the hydrogens that are released here. All the hydrogens will go through the same process. They will be broken down into H plus and electron. Electron will go through the electron transport chain, release energy. This energy is used to transport H plus out of the inner membrane. And the H plus will gather here. Hydrogen plus ions will be accumulated in the intermembranal space. That is why this part is low in pH. Okay, the intermembranal space has low pH because of the accumulation of H plus ions. Now a steep gradient is created. For mitochondria, you should know that the outer membrane and the inner membrane, they have different permeability. Both are selectively permeable, but the inner membrane is highly selective. Okay, the inner membrane of mitochondria is highly selective, so it won't let H plus get back in. H plus is going out of the inner membrane, but it cannot come back in. Though it wants to, because there is a steep gradient, H plus would die to come in, but it is not allowed by the inner membrane. So H plus will be accumulated in the intermembranal space, and then one thing happens. There is a channel protein that opens up to let the hydrogen move back into the mitochondrial matrix. So as the channel protein opens, it has an enzyme here which is called ATP synthase. Now we have learned about two enzymes here. ATPase is the enzyme, or ATPase is the enzyme that breaks down ATP and releases energy. The reverse of which happens here. ATP synthase. Do not confuse ATP synthase and ATPase. ATP synthase is the one that basically combines adenosine diphosphate ADP and inorganic phosphate to make ATP. That's what will happen here. The hydrogen that is moving here, it's positively charged. And when the charge is flowing, it has the electrical potential. And this electrical potential is being used by this ATP synthase to join ADP and PI and make ATP. Finally, the H plus will accept its electron back from the electron transport chain. It will react with oxygen. Oxygen is the final acceptor. If the oxygen is not present, all of these reactions will not happen. So oxygen must be there to act as the final acceptor of hydrogen. So oxygen accepts the hydrogen as the final acceptor and water is formed. Oxidative phosphorylation is concluded here. What is happening? Reduced NAD and reduced FAD will lose their hydrogen at the site of inner membrane. Hydrogen will be broken down into proton and electron. Electron goes to electron transport chain, releases energy, and this energy is used to transport proton out of the inner membrane, which gets accumulated in the intermembranal space. It comes back in through the channel. Finally, this electrical potential is used to make ATP. Okay, hydrogen is accepted by the oxygen, and finally water is formed. Now, if we have to make a conclusion of how much energy we have made through all these processes, if we go back to the first step, glycolysis, we can see that there is net gain of 2 ATP molecules. So 2 ATP are formed by glycolysis. If we go to the link reaction, there is no ATP being formed here. No ATP formed during link reaction. Krebs cycle. If we look at this process, we will only see 1 ATP, but because there are 2 pyruvate molecules, so we would consider that by the oxidation of glucose, 1 glucose molecule, there are 2 Krebs cycles happening, and because of 2 Krebs cycles happening, we will consider that 2 ATP molecules are formed. So 2 ATP molecules in glycolysis, 2 ATP molecules in Krebs cycle, and finally oxidative phosphorylation, because there is electrical potential. The highest number of ATP molecules, 28 ATP molecules are formed here. In total, we have 32 ATP molecules formed just by the oxidation of 1 glucose molecule. In the next step, we are going to learn anaerobic respiration. In anaerobic respiration, only glycolysis is happening. Now, you would consider oxidative phosphorylation is the only step where oxygen is used. There is no oxygen in Krebs cycle, there is no oxygen in link reaction. So why would the link reaction stop in the absence of oxygen? Why would the Krebs cycle stop in the absence of oxygen? So we will learn that, and we will learn how energy is formed by glycolysis. A small amount, however, of energy is formed during anaerobic respiration in muscles or maybe in living organisms like yeast. Also, we will also see that glucose is not the only respiratory substrate that our body uses, or carbohydrate is not the only respiratory substrate. Though given the choice, our body would always prefer using glucose as respiratory substrate, but lipids can also be used, and they release even more energy, more than double of the energy of glucose. And proteins can also be used, and they also release energy more than carbohydrates. Both proteins and lipids are releasing more energy, but our body would always prefer glucose. To answer all those questions, we will be back with a new video, so stay connected and subscribe our channel to keep yourself updated. Thank you very much.